genome sequencing has increasingly become an important tool for studying disease outbreaks. The SARS-CoV-2 genome used in this study was sequenced from a sample of bronchoalveolar lavage fluid from a single patient who was worker at the market, and who was admitted to the Central Hospital of Wuhan on 26 December 2019. Now let's learn how to download the virus from NCBI database. NCBI, or National Center for Biotechnology Information, is a branch of the U.S. National Library of Medicine. It's a hub for biological and biomedical data, including DNA and protein sequences, scientific publications, and genomic information. Researchers use NCBI for tasks like finding DNA similarities, accessing scientific papers, and exploring genome data. It's a vital resource for advancing genetics, genomics, and related fields. Now let's download our file that we will analyze. In step 1, we open Google and we write NCBI. NCBI. Now let's log into the website. Then, as we can see, we can search in many different databases. Like here we have all databases, Simply, Biocollections, Homologene, Protein, and a lot of databases. Today we are interested in Nucleotide database, so we choose Nucleotide. The Nucleotide database in NCBI is a comprehensive collection of nucleic acid sequences, including DNA and RNA sequences. It serves as a repository for genetic information from various organisms, including genes, genomes, and genetic elements. The Nucleotide database is a compilation of genetic sequences derived from various sources, such as GenBank, RefSeq, TPA, and PDB. These sequences include genome, gene, and transcript data, which serve as fundamental resources for biomedical research and facilitate discoveries in the field of genetics and genomics. So we choose Nucleotide, then we write the name of our virus. We write respiratory syndrome coronavirus 2. Respiratory syndrome coronavirus 2. And enter. We choose the first option. And here we have it. Severe acute respiratory syndrome coronavirus 2. Now let's download our FASTA file. And from Send to, we choose File, and then Format FASTA. Now let's click on Create File. Here we have it, our FASTA file. Before we move on, let's try to understand what's here. Locus NC045512. This line provides basic information about the sequence, including its accession number and characteristics. In this case, it's a single-stranded RNA molecule with a length of 29,903 base pairs, representing the complete genome of severe acute respiratory syndrome coronavirus 2. Regarding definition, it briefly describes the sequence, mentioning its complete genome of SARS-CoV-2 from the Wuhan HU1 isolate. Accession NC045512. This is the accession number assigned to this sequence in the database. Version NC045512.2 indicates the version of this sequence. DB link provides a link to a related body project. Keywords RefSeq indicates that this sequence is part of the reference sequence collection. Source specifies the organism that this sequence comes from, in this case SARS-CoV-2. Reference lists references to specific publications related to this sequence, including the authors, titled journals, and PubMed's IDs. Comment. Additional information such as how this record has been curated, updates to the sequence, and links to protein structures and related sequences. And finally, let's go up. As we can see here, there is FASTA. If we click on it, then we are able to see our sequence. Here all our sequence. Now let's begin our analysis. Greetings everyone. In this video we are going to address COVID-19 virus or SARS-CoV-2. We will start by reading a FASTA file, but before we read it, we need to upload the libraries that we are going to use to read our file and make our analysis. It's like wearing glasses before we start to read. Now let's load our file that contains the sequences from the FASTA file. FASTA _ file. Now let's print the genome sequence variable that we created. As we can see, we got ID, name, description, number of features, and sequence. For the ID NC045512.2, this line displays the identifier or ID of the sequence, which is usually derived from the header line in the FASTA file. In this case, the ID is NC045512.2, and for name NC045512.2 also, the name line also repeats the identifier for the sequence. In FASTA files, the ID and name often contain the same information. This redundancy is a common practice to ensure that users can easily access and identify the sequence by either the ID or the name. The choice to include both an ID or name is often made to provide users with flexibility in how they reference or search for sequences. For example, some users might prefer to reference sequences by their short and concise IDs, especially when working with large datasets or scripts. Others might find it more informative to use the name, which can contain additional details about the sequences, such as the organism, strain, or specific information about the sequence origin or purpose. And for description, NC045512.2, severe acute respiratory syndrome coronavirus to isolate Wuhan, HU1. This line provides a longer description of the sequence. It typically includes additional details about the sequence, such as the organism it belongs to, its isolate name Wuhan HU1 in this case, and its status as complete genome. This description is useful for understanding the content and origin of the sequence. And for number of features equals 0, This line indicates that there is no annotated features or regions with specific information, such as genes or coding regions associated with the sequence. And for sequence, this is the actual DNA sequence represented as BioPython sequence. It starts with ATT, AAA, GG, TTT, and ends with AAA. This is the primary sequence data that we will analyze and manipulate in our Python. Now let's determine the length of our sequence. Sequence length equal to then genome sequence and sequence length. We got 29,903, and it is the same numbers that we found on the NCBI website. In this case, it's a single-stranded RNA molecule with a length of 29,903 base pairs. Now let's access the DNA sequence from the seq-record object. So DNA sequence equal to genome sequence dot seq. Now let's take the first 30 characters from our DNA sequence, and then let's perform transcription, and we take the first 30 characters of our RNA. After that, let's take the first 10 proteins. So first 30 DNA equal to DNA underscore sequence and then 30. Now print first 30 DNA. Here we have it, the first 30 characters in our DNA sequence, starting from ATT, AAA, GGG, and ends with CAA. Now let's take the first 30 RNA character. So first 30 RNA equal to DNA underscore sequence dot transcribe to make transcription process, then 30 and first 30 RNA. And here we have it. Our DNA sequence is converted to RNA. As we said before, in RNA there is no thymines. So A is A, and T, which is thymine, is converted to uracil. So T is U again, and AAA is the same, and GG the same also. 3 thymines converted to 3 uracils, and so on. Now let's get the first 10 proteins. So first 10 proteins. Near underscore sequence dot translate. As we know, the mRNA is converted to the proteins after the translation process. Goes on, which you stand. Now print our proteins. And here we have it, our 10 proteins. Great, but what this asterisk or star line mean? In the context of a protein sequence, the asterisk or star sign represents a stop codon or termination codon. Stop codons are special sequences in the genetic code that signal the end of protein translation during protein synthesis. They don't code for an amino acid, and their presence indicates that the protein is complete. DNA and RNA are both composed of nucleotides, which are the building blocks of genetic material. In DNA, adenine, or A, always pairs with thymine, and cytosine C always pairs with guanine G. In RNA, adenine A still pairs with uracil U, but the presence of thymine T is replaced with uracil. This base pairing specificity ensures that the information is accurately transcribed from DNA to RNA. Thymine and uracils are structurally similar, with thymine having a methyl group CH3 that uracil lacks. This methyl group in thymine is not presented in uracil. The absence of methyl group in uracil simplifies the chemical structure and is one reason for the substitution. This simplification is advantageous for the RNA molecule because it makes RNA more flexible and less stable than DNA. RNA is often involved in transient processes and doesn't need the long-term stability that DNA provides. Great. Then, as we mentioned before, in our case, our sequence has 29,903 nucleotide bases. But how many G nucleotides we have? Or how many C or A or T? Let's count our nucleotides by making a list comprehension and a for loop inside it. We did a for loop video explaining how to make for loops in Python. You can check it out from the notification that appears on the screen now. Now let's get started. Count nucleotides. Equal to A, T, J, and C. In counts equal to sequence of seq dot count base for base in nucleotides. And then let's make our for loop. I is in at is types of Now let's print the counts for each nucleotide. And here we have it. The number of A or adenine is 8,954, and the number of T, thymine, is 9,594, and for G's are 5,863, and lastly, the cytosines are 5,492 nucleotides. Now let's break down our code. Firstly, for nucleotides ATGC, here we created a list named nucleotides containing the 4 letters or bases. These bases represent the 4 types of our nucleotides in the DNA: adenine, thymine, guanine, and cytosine, and then counts genome sequence.seq.count base for base in nucleotides. Here we created another list called counts. It counts how many times each of the 4 nucleotides appears in the genome sequence. For each base, we put the letter in the nucleotides list. It counts how many times that base appears in the genome sequence. So it counts the number of A's, T's, G's, and C's in the sequence. The counts are stored in the counts list in the same order as the nucleotides list. If you are interested to watch our Python video, which is only about list comprehension, then please check it out for notification that appears on the screen now. And then for I base in enumerate nucleotides, here we set up a loop that goes through each element in nucleotides list one by one. It also keeps track of the position index of each element using I. The enumerate function is used to get both the index I and the nucleotide base from the list. And then print the number of base and then counts, both between curly brackets, and then I besides the counts. Here we print the counts of each nucleotide along with its name. We used f-string to format the output. So for each nucleotide in the loop, it will display a message like the number of A, for example, in our case is 8954, where A is the nucleotide name and 8954 is the count. To have a better idea and to make it easier to understand, let's plot our nucleotides. So let's create a bar chart using the counts list. In step 1, we set the figure size. Dot. Step 1. Select figure size. Plot.figure. X size equal to 12.5. Here we set the width and height, and for step 2 we choose the type of the figure. In our case we perform a bar chart. Now step 2, choose the type of the chart. Plot bar, nucleotides, counts, here we choose X and Y, color, within the width, bracket 0.6. Now we set the X and Y labels. So plot.x label, we make it nucleotide. Plot.y label, we make it count. Plot.title, makes a title. Nucleotide counts, and lastly plot.show. And here we have it. As we can see, it's more obvious now that the T's are the most nucleotide in the sequence, followed by A, followed by G, and the least nucleotides are the cytosines or C's. But what if we want to make the value of each nucleotide appears on each bar, so it's visually better? In this case, we will take the above code, copy and paste. Then we will make a for loop. So we take it here, and now let's make our for loop for I enumerate counts plot text I count or count center, bottom, black. And then plot dot show. Here we got an error because we wrote plot dot show twice, so we'll have to delete this. And here we have it, our plot, but with the number of each nucleotide at the top of the bar of each nucleotide. So here we have the number of adenine, which is 1,954 nucleotides. We got also the same for thymines and guanines and cytosines. Now let's break down the loop step by step. So for I count in enumerate counts, here we started a for loop that iterates through counts list. This contains the counts of different nucleotides. The enumerate function is used to get both the index I and the value count from the list as we iterate through it. I represents the index of the current nucleotide, 0, 1, 2, etc. And count represents the count of that nucleotide. Plot.text, I count, str count, and then HA equal center, VA equal bottom, color equal black. Here we added a text label to the bar chart. I is the x coordinate where the label will be placed, and it corresponds to the index of the nucleotide in the list. This positions the label at the center of the corresponding bar. Count is the y coordinate where the label will be placed, and it represents the count of the nucleotide. This positions the label on the top of the bar at the height corresponding to the count. str count converts the count to a string, so that it can be displayed as text on the chart. HA equals center. This specifies the horizontal alignment of the text should be centered at the x coordinate. And VA equal bottom specifies the vertical alignment of the text should be at the bottom, meaning the text is anchored at the bottom of the count value. And then we set the color equal to black. Now let's talk about the GC content. GC content, short for guanine cytosine content, is the percentage of bases in a DNA sequence that are either guanine or cytosine. GC content is a fundamental metric in genomics and molecular biology. It can provide insights into various aspects of DNA sequence, including its stability, melting temperature, and potential protein coding regions. Differences in GC content can also be indicative of different genomic regions or organisms. But what are the applications of GC content? GC content is often used to identify potential protein coding regions, regulatory elements, or other fundamental elements within a genome. It can also be used for taxonomic classification and comparative genomics. Now let's calculate the GC content. GC underscore percentage equal to ET percentage in print GC content As you can see, we got the GC content and the AT content. The percentage for the GC content is 37.9%, whereas the percentage for AT content is 62%. Regarding GC underscore count equals sum, one for base in genome underscore sequence dot seq if base in GC. For each character base in genome sequence dot seq, Python checks if the base is either G or C. If the base is G or C, it adds 1 to the GC count. In the end, GC count will hold the total count of G and C bases in the sequence. So we use a generator expression within sum, one for base in genome sequence to seek if base in GC. We use it to iterate through each base in the sequence, if base in GC, checking if it's either G or C if base in GC. For each matching base, it generates a value of 1. Then, by some function, it adds up all these 1 values, effectively counting the total number of G and C bases in the sequence. This count is then stored in the variable GC count. And for GC percentage, it calculates the percentage of GC count in the given sequence. It divides the count of GC bases in GC count by the total length of the sequence underscore length. This gives us the fraction of bases that are GC. Then it multiplies this fraction by 100 to convert it into percentage. And the same for 80%. We can also calculate the GC content by using the GC fraction function. So GC underscore count equal to GC underscore fraction DNA underscore sequence. Then print GC underscore count from Bioinformatics import GC underscore fraction. As you can see, we got the same percentage, and then we multiply it by 100%. As you can see, we got the same percentage, which is 33.9. Now let's go for the GC skew. GC skew is when the nucleotides guanine and cytosines are over or under abundant in a particular region of DNA or RNA. GC skew is also a statistical method for measuring strand-specific guanine overrepresentation in equilibrium conditions without mutational or selective pressure and with nucleotide randomly distributed within the genome. There is an equal frequency of the 4 DNA bases, adenine, guanine, thymine, and cytosine, on both single strands of a DNA molecule. However, in most bacteria, for example like Escherichia coli and some Anarachia, like Sulfateraceous, nucleotide compositions are asymmetric between the leading strand and the lagging strand. As we know, the leading strand contains more guanine and thymine, whereas the lagging strand contains more adenine and cytosine. This phenomenon is referred to as GC skew equal to G minus C over G plus C, whereas G represents the guanine nucleotides in the DNA sequence and C represents the cytosine nucleotides in the DNA sequence. Now let's calculate the GC skew. Firstly, we determine the nucleotide bases, which are A, T, G, and C. And then we determine the number of all bases inside our sequence. increase in nucleotides. Then we extract the counts for G and C. Counts and C count, take a copy and paste and make it C instead of G. Now we calculate the G's skew. As we said before, the GC skew equal to G count minus C count over G count plus C count. So G square count minus C square count over G count plus C count. Now print GC skew. As you can see, we got the GC skew, which is 0.032. The calculation of G count minus C count over G count plus C count, it calculates the overall G skew for the entire DNA sequence, considering the total counts of G and C nucleotides in the sequence. So as we mentioned before, G count represents the total count of guanine or G, and C count represents the total count of cytosine or C. The formula calculates the GC skew for the entire sequence by taking the difference between the total counts of G and C and then dividing it by the sum of the total counts of G and C. The numeric value indicates the degree and direction of skew in the nucleotide composition of the DNA sequence. Specifically, in this case, the positive value suggests that there is a slight excess of guanine nucleotide over cytosine. The magnitude of the value represents the extent of the skew. A larger positive value would indicate a more pronounced skew towards G, while a negative value would indicate a skew towards C. A value of 0 would indicate an equal proportion of G and C nucleotides. Alright, now let's create a GC skew plot. But how do we do it? We are going to calculate the GC skew for every position along the DNA sequence. GC skew is a measure of the balance between guanine and cytosine nucleotides at different points in the sequence. By calculating the GC skew at each position, we can create a plot that shows us how this balance changes along the sequence. For each position, we will look at the basis and determine if there is a bias towards more G's or more C's. Positive values indicate an excess of G's, while negative values suggest an excess of C's. By plotting this information for every point in the sequence, we can visualize variations in the GC skew. These variations often correspond to important genomic features. For example, shifts in the GC skew can signal the location of the DNA replication origin, a crucial event in the biology of the organism. Let's take a look at a small DNA sequence to better understand the GC skew. Then we implement it on our large sequence. Imagine we have the following sequence: ATG, CAG, CTT, AGCG. We want to calculate the GC skew at each position along this sequence. We will move a small window, let's say a window of 5 bases, along the sequence, and for each window we will count the number of G's and C's. For example, at the start, our window covers the sequence ATGCA. If we count, we find that there is 1 G and 1 C. So the GC skew at this position would be 1 minus 1 over 1 plus 1 equal to 0. We then slide the window 1 base into the right, covering TGCAG. Now we have 2 G's and 1 C. So the GC skew at this position would be 2 minus 1 over 2 plus 1 equal to 0.33. We repeat this process for every position, and at the end we will have a series of GC skew values that tells us how the balance of G and C nucleotide changes along the sequence. In our plot, we will see each point representing a position in the sequence, and the vertical position of the point shows GC skew at that position. Positive values indicate a bias toward G, and negative values indicate a bias towards C. Now let's write our code. Firstly, we import our libraries. Then we load our FASTA file. Then we create the sequence required. Then we get the DNA sequence. Then we calculate the GC skew by making a function calculate GC skew. Then we avoid the division by 0 by checking if both counts are 0. So if G underscore count plus C underscore count equal 0, then GC skew dot append 0. Else now we return the GC skew. Then we calculate the GC skew values. The name of our function. Then we put the sequence. Now we print the GC skews, but we will print only the first 50 values in order to give us an idea how the GC values looks like. And now we create the plot. plot.py and now we show the plot. Here we have it. These are the GC skew values, and here is the GC skew plot. Firstly, let's explain the code. We created a function, calculate GC skew and sequence inside. We define a function called GC skew that takes a DNA sequence as input and calculates the GC skew. Inside the calculate GC skew function, GC skew is initialized as an empty list to store the GC skew values. G-count and C-count are initialized to 0 to keep track of counts of G and C nucleotides respectively. The code then iterates through each base in the DNA sequence. If the base is G, GC count is incremented by 1. If it's C, C count is incremented by 1. To avoid division by 0 error, it checks if both G count and C count are 0. If so, it appends 0 to the GC skew. Otherwise, it calculates the GC skew using the formula G count minus C count over G count plus C count, and appends the result to GC skew. GC skew values equal calculate GC skew and the sequence inside. Here we call the calculate GC skew function with the DNA sequence and stores the resulting GC skew values in the GC skew values list. Then we print the first 50 values of the GC skew, and we plot the figure by setting the figure size, width equal 10 and height equal 6. Then we plot range length sequence. GC skew values label equal GC skew and color equal blue. Here we created the actual GC skew plot. It uses the range length sequence as the x-axis position in the sequence, and GC skew values as the y-axis, which is GC skew values. Then we set the x label, y label, and title. Then we add the horizontal dashed line GC skew value equals 0. Now let's talk about the chart. The y-axis represents the GC skew values, which are calculated as the difference between the count of guanine nucleotide and the count of cytosine nucleotides, divided by their sum. The scale on the y-axis ranges from approximately negative 0.25 to 0.1. The x-axis represents the position in the DNA sequence, ranging from 0 to around 30,000. Assuming the sequence is 30,000 base pairs long, this axis essentially shows us the location or position within the DNA sequence. The plot starts as position 0 on the x-axis and negative 0.25 on the y-axis. This indicates that at the beginning of the sequence, there is a skew towards more cytosine C than guanine G. This skew is reflected in the negative GC skew value as we move along the sequence from left to right on the x-axis. The GC skew value starts to increase, reaching its maximum value of approximately 0.1. This increase in the G skew suggests that there is an increasing imbalance in the counts of G and C as we progress along the sequence. This could be indicative of a region where there are more Gs relative to Cs. That's for today, but we are not done yet. In the next video, we will be diving even deeper. Stay tuned for part 2. Thanks for watching. I hope you enjoyed the video. If yes, then please don't forget to subscribe to our channel and hit the like button. Until next time, happy analyzing.